Hello everyone, my name is Ashfaq Ahmed and I am working in the field of bioinformatics in structural biology. In our last video, I taught you how to generate an overall view of the inhibitor and protein using PyMOL. You can generate this view from a PDB structure or for the structure you docked using any program. Today we will learn how to analyze specific interaction in PyMOL. In our last video, I did not discuss the analysis and visualization of specific interaction. Let's say hydrogen bond, let's say metallic interaction, let's say van der Waals interactions, or any other interaction, for example pi staking. So in this video we will analyze such kind of information, and also we will visualize using the same program PyMOL. So let's start it. In today's session we will use two things. We will use PLIP, that stands for Protein-Ligand Interaction Profiler, and we will use PyMOL. PLIP is currently state-of-the-art algorithm, or you can say software, which calculates almost all sort of interactions for a protein-ligand complex or for a protein inhibitor complex. Please do not confuse yourself with the word inhibitor, agonist, antagonist in ligands, or compounds, or drugs. So one can use a word according to their research project. So here I will use a word ligand. It's a general word and almost acceptable for everyone. So let's start. Okay. For today's tutorial session, I will use a PDB structure 603R. 603R, it's open. If you don't know how to open such kind of structure, let's say, let me repeat it for you. This is RCSB. You need to type the code 630R and just give me time. Wait. (603R). Okay, this is a structure. Then here you can see the download file option. Just click here and then click this one, PDB format. Once you click here, the structure will be downloaded into your download folder. Here it is. Now the second thing is you have to write down, let's say write PLIP, PLIP for Google, and you will see this thing, Protein-Ligand Interaction Profiler. If you use this program, you need to cite the research paper or their paper. So this is the PLIP research paper, so you need to cite this paper in your research paper or thesis, whatever. Okay, so open it. Okay, how you will use PLIP? There are two options. For a PDB structures, you can simply write the code here. You can see here, find PDB ID using our search tool. That's okay. But in case you have docked your own structure, and which is not present in the PDB, or it is present in the PDB but without that specific ligand. So in both case you will upload your structure manually. So in this case I have both the options because I downloaded the structure from PDB, so I can use this option, choose file, and then from download select the 603R, or I can use directly from here 603R. So it's up to me, also it's up to you which panels to use, the left one or the right one. Let's say use this one. It will take almost 30 to 40 seconds, and you will have this kind of stuff. You can see. Okay, now do not make yourself in a hurry. So if you see, the result contain two things. It's a ion and it says small molecule plus ion. So if you selected this result, it will not be wrong, but it will be incomplete. And remember, you will see the ion interaction only for those protein if it contains some ions. In this case 603R contains 2 zinc ion. You can see zinc. So therefore the program splits the ionic interactions in a separate panel, and if you are interested in your ligand interaction, then they are here. Okay, so if you see, these are the amino acids, this is the methylene. You can see here from the color methylene, and these are actually the water bridges because they are also making some water bridges. And what kind of interaction they are making, for example this one, this color, it's making pi stacking, and this one, they are the hydrophobic interaction. And this program will visualize or will tell about the hydrogen interaction and all these interaction if they are present in some specific kind of representation and some different colors. Okay, but how to import these things into PyMOL, and then how to make a beautiful picture from these such kind of analysis or from such kind of informations. So if you see here, there are two options: download visualization in PyMOL format, download visualization as image. I will not suggest you to download directly this image as a PNG. However, you can, if you think that this scene is perfect for you, just download it and you can use it. If you want to see the visuals in 3D here in your browser, so just click here. You can also see directly here. Okay, if you want to include some information in table form, then you have these tables, just like hydrophobic interactions, these two. This is the index number. In this figure we have two hydrophobic interactions. One is 67, this is the chain number A. Other one is 73. On 67 it is methionine, on 73 it is valine. The hydrophobic interaction are formed in the range of this distance. It is in angstrom. Which number of atom is used from the ligand side? So this one, and from the protein side is this one. Okay, there are 3 water bridges, so this is the information. one pi is taking, this is the information, okay, and it makes four interactions. I will call it metallic interactions or metal interaction or ionic interactions. It means the interaction of amino acids with metal ions. So these are the metal complexes are metal interactions, okay. Now in this structure we do not see any hydrogen bond, so it's okay, you will see it for your, I'm not seeing in this figure, so that's okay. Okay, now I will download this one, download visualization in PyMOL format. So download it, it will be downloaded here. Now I have already opened a PyMOL. You can see here I have a PyMOL. So I will teach you now how you can, before going to open the structure, we need some adjustments. What adjustments? We have already this structure downloaded. We need to first open the structure 603R. Okay, it is open here. Now what you need to do, just click here, let's say the A button, action button, and say remove water. Okay, now click here zinc, and this is the compound ID or ligand ID XJE. Okay, these three things are selected, you can see, and delete it, remove atom. Okay, now I made a clean copy of only protein, and now file, export molecule, and here you say enable current, it's okay, save. Here you need to select the PDB option because we want to select it in the PDB format, 603R-1. okay, I name it this way, this is PDB, and save it in the download folder. Okay, now we have two files, in fact we have three files in our download folder. The first one we have is 603R, which we downloaded from the PDB, or you may have from your docking assignment. This is file number 1. We have second file 603R-1, which we have generated just now. It is almost the same file with 603R, but this one do not contain the zinc atoms, do not contains the ligand, and do not contains water molecule. And this, the third file, 603R_XJE_A_303PSE. So actually this is the file we downloaded from here, because if you see its extension is PSE. So we are now we will open this PSE file. So file, open. Okay, now you can see the same thing mentioned here. Why I discourage you that do not use this direct PNG, because I want you to manipulate the scene according to your own requirement. Maybe I like this pose, maybe someone like this pose, maybe someone like different colors, maybe someone like some different kind of representation and surface and each and everything. So PyMOL is a very flexible program, and I hope you have learned too much things here in PyMOL. but today I will show you some more stuff here. Okay, now we have opened this thing. The second thing, now we need to, if you are okay with this structure, so you can just do some changes here. For example, I click here, it is selected here, and then say show spheres silly. Silly means for selection here. So we have used this command also in our previous video. So show sphere for selection, enter. You can see. Now if you want the spheres to be bigger, then you say set sphere underscore scale comma 0. Let's say 35. Oh wait, okay, it is already in 0.35. make it let's say we are going to make it bigger. Okay, so this is sphere for the ligand. Now I'm moving to show spheres for these things, so you will need to select all these residues. Let's say I have selected. Now in this silly option or the selection option, you have these residues. Okay, show sphere silly, you can see, and make the sphere scale just like 20, maybe just like 30. Okay, now if you want some different colors, let's say I want these colors to be red or anything else. So let's say I have again selected all these things, and now color, let's say I'm selecting this one, or maybe magenta pink. Okay, in short, you can do whatever you want. If you want to generate some surface here, so it is also easy from show option and generate surface, spheres, mesh, dots, whatever you want. Okay, now from here, display background white, that is already white. Quality, maximum quality, you need to select it. Okay, I have selected. So the final option is to generate your figure as we did in our last video. But hold on, I'm going to make this figure more interesting. How? See, I'm going to open another file we have prepared, that is 603r underscore dash 1. Open. Now you can see, now you understand that why I had removed these things, the ions and the water and other things from that structure, because I don't want to make some doublings. Okay, now these things, the helices are in the fancy shape. If you don't like this fancy shape, just come to setting here cartoon and uncheck this fancy helices. It's become this. Now what I like, normally I make these helices a bit slimmer, so set cartoon all length 0.7. can you see now they're slimmer? And for set cartoon all instead of length you say width, and I'm typing this thing. Okay, now if you want to make a surface for this figure, that will also be interesting. Or if you don't want a surface, so that is also okay. Let's say I'm trying for a surface. How will it look like surface? But remember, the surface will be with for 603R-1. But the problem with the surface is that it has almost disappeared some of the regions. So here we have also another option for set transparency underscore scale 0.2. we have used this in our last video. Okay, I think we don't need this scale. No, okay, now you can see something in the transparency, that is 0.2. let me increase it to 0.7. now see. Okay, maybe I want to change this color to a uniform color which may match to the okay. So this is your figure. Let me make it now. That how will it look like? Wait, let's say this one. Okay, now ray I'm using 2000-2000. once again I'm going to tell you, this is for the length of the figure and this is for height of the figure. Okay, I have entered it. Now it may take 1 or 2 minutes, so therefore I'm going to pause my video for that time. So here it is, our figure. You can see. Now I will try to save it, but where to save it? So last time I have also taught you this method. For example, I am interested to save it here, maybe. Okay, let me teach you. This is a folder on my desktop 1 2 3. Let's say I'm interested here. So you need to open this folder, then click here, copy this path. Now write CD and then Control V to paste the path. Now you can see, now CD, it says now in C. This is the complete address of the folder. So now you need to type PNG and give it some name. Let's say I'm giving name PLIP dot PNG. Enter. Okay, when it says wrote this amount of pixel image to the file PLIP.png. Okay, I'm going to close it. Now let's see what kind of figure we have generated. Oh, it should be this one. Wait, PLIP. Okay, this is our figure, and I will also use it for thumbnail, so you can also see it on the main screen. And these are the interactions, and what kind of interaction, what does it mean. All these informations are present here. Also they are present here. If you see the interaction, what are the metallic interaction. For example, you click here. Once you click, it will tell you like this is cysteine 208, and the interacting atom is carbon beta, and it is present in chain A, and it is present in this protein 603R. So the same way, if you need the information of this amino acid, click here. It will tell you this is in chain A, this is valine, this is 73, and the interaction is on carbon beta. Okay, so the same thing you can do with other residues, and finally you will need to label it by any program of your choice. Normally I'm using Canvas or Paint.net. It is up to you which program you want to use it. So I hope you have liked this, you like this video, and if you are enjoying this channel, so subscribe and share these information with all other colleagues. If you are interested in some specific tutorial video or some specific topic, so you can drop your question or your request in the comment section of any video you are watching. I will try my best to make some tutorial or some fruitful video on this topic. So that's enough for today. Bye bye, stay happy.